Assalamualaikum warahmatullahi wabarakatuh. We are from Group 8. Today we are going to explain about detection of SARS-CoV-2 through real-time reverse transcription polymerase chain reaction. The content of the presentation is the principle behind the technique, the method or process involved, the advantages of the technique and the limitations of the technique. The principle of the technique is in RT-PCR, the RNA template is first converted into a complementary DNA, cDNA, using a reverse transcriptase. The cDNA is then used as template for exponential amplification using PCR. There are two types of PCR. The first one is one-step PCR and the next is two-step PCR. In the one-step approach, the entire reaction occurs in a single tube. In two-step reaction, the reverse transcriptase reaction and PCR amplification will be performed in separate tubes. There are protocols in both types of PCR. The protocol in one-step RT-PCR select a one-step RT-PCR kit which should include a mix with reverse transcriptase and the PCR system such as tag DNA polymerase and a proofreading polymerase. Prepare a reaction mix which will include deoxynucleoside triphosphates, primers, template RNA, necessary enzyme, and a buffer solution. Add the mix to a PCR tube for each reaction, then add the template RNA. Place PCR tubes in the thermocycler to begin cycling. The first cycle is reverse transcription to synthesize the cDNA. The second cycle is initial denaturation. During this cycle, reverse transcriptase is inactivated. The next 40 to 50 cycles are the amplification program, which consists of three steps, which are denaturation, annealing, and elongation. The RT-PCR products can then be analyzed with gel electrophoresis. Protocols for two-step PCR. Step 1. Combine template RNA primer, the NTP mix, and nucleus-free water in a PCR tube. Add RNase inhibitor and reverse transcriptase to the PCR tube. Place PCR tube in thermal cycler for one cycle that includes annealing, extending, and then inactivating reverse transcriptase. Proceed directly to PCR or store on ice until PCR can be performed. Step 2. Add a master mix containing buffer, the NTP mix, MgCl2, tag polymerase, and nucleus-free water to each PCR tube. Add appropriate primer. Place PCR tubes in thermocycler for 30 cycles of the amplification program, which includes three steps, which are denaturation, annealing, and elongation. The RT-PCR products can then be analyzed with gel electrophore contamination due to more frequent sample handling.
For the method, the sample will be collected by nasopharyngeal swab or oropharyngeal swab. The swab is immediately placed into a sterile tube containing a viral transport medium. PCR is used to make millions to billions of copies of a specific DNA fragment rapidly. RNA in virus must be converted into complementary DNA sequence by reverse transcription to continue with PCR. To start the PCR, the sample is first added into microcentrifuge tube, then mixed with lysis buffer. This buffer consists of phenol and guanidate isothiazuride. Once the buffer is added, the tube is mixed and incubated at room temperature. The virus sliced by the lysis buffer. Once sliced, purification procedure carried out by using a spin column, sample loaded onto spin column, then centrifugation is performed. RNA molecules are bind to the silica gel membrane. Protein and other contaminants are not retained. Then, wash buffer is added and column is put into centrifuge again. This removes any remaining impurities from the membrane lining leaving only RNA to the silica gel. Once the sample is washed, the elution buffer is added. Elution buffer removes viral RNA from spin column. Then we will get the viral RNA. Then master mix is used. Master mix consists of buffer, RT enzyme, nucleotide, fold, primer, tagman prop, and DNA polymerase. To complete this reaction, RNA template is added into master mix and mixed. Mixture is then loaded into PCR plate and will be placed in a PCR machine. In the PCR machine, choice of target gene depends on the primers and prop sequence. The first step in RT-PCR is reverse-transcription. First-strand complementary DNA synthesis is primed with PCR reverse primer. Reverse transcriptase will then add DNA nucleotide into 3' end. Next, initial denaturation is applied, causing denaturation of RNA-DNA hybrid. This step is required for the activation of DNA polymerase. PCR consists of series of thermal cycles which are denaturation, annealing, and extension step. This step is for denaturation of double-strand DNA template that happen at 95 degrees Celsius. Then the annealing process will be at 58 degrees Celsius. Extension step, DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand. Then, in cycle 2, denaturation is performed, yielding 2 single-stranded DNA molecules. The temperature is lowered, allowing annealing process. Annealing of the tagman prop to its complementary part of the target DNA. Tagman prop consists of reporter of fluorescence that emitted by the fluorophore when excited by the cycler's light source and quencher. In the extension step, the DNA polymerase synthesizes new strand. When polymerase reaches tagman prop, the dye will be separated from the quencher. Then, the cycle will be repeated. The number of double-stranded DNA pieces is doubled in each cycle. Therefore, PCR can be used to analyze extremely small amounts of sample. This is the pathway for the measurement of fluorescence signal. In each PCR cycle, light from excited fluorophore can be detected by the CCD camera, which convert the light that is captured into digital data. This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction as it occurs in real time.
The advantages of using RT-PCR technique are: the first one is high quantitative and specificity. Absolute copy number and gene expression fold changes can be measured accurately. Closed tube format eliminates risk of cross-contamination. Second one is high sensitivity. It allows detection of a low copy target with greater confidence and very wide dynamic range, which are 1 to 10 copies detectable within a single run. The next one is RT-PCR is faster because no agarose gels were needed, and it is also safe because no ethidium bromide or radioactive needed. The last advantage is rapid detection.
There are a few limitations in using the real-time reverse transcription polymerase chain reaction, such as it is high cost, which is very expensive due to the use of expensive consumables. Next, the increase of false negative and false positive in pathogen detection, especially for new emerging or highly variable pathogens. Next is medium turnaround time, restriction on sample transportation, and it needs for instruction. That's all from us. Thank you.